So we'll be looking at randomized trial. Okay. So we'll quickly run through randomized trial and different phases for a clinical trial. Then, if anybody have a question, you look at that, because today will be our last. So this is what we discussed last week. We have looked at experimentation of the randomized trial. So we're looking at the experimental studies. So we say that the randomized trial, we look at the allocation, randomized allocation of the study subject into a treatment and control group. So that's what you do. So we look at the outcome stroke, the endpoint you are looking at. The outcome and endpoint can be improvement or the desired result effect. So you look at the effect. There's also a side effect because every drug has a side effect. So yes, you look at the desired clinical outcome you want, but there is also a side effect that can result as a result of a biological or unwanted interaction with the active molecule, whatever active molecule that you are given the control group. So we talked about blinding. In blinding simply means deny information of the treatment and control sections. So in blinding, you are making the people that are taking the drug, they don't know what they are taking. Or you can do it in such a way, we call it, you have single blinding, you have double blinding, you have triple blinding. So in single blinding, the people taking the drug don't know what they are taking. In double blinding, the subject and the investigators, maybe the doctors, the nurses, the pharmacists, they don't know what they are given, and the people taking the drug don't know what they are taking. Then triple blinding, the subject, the investigators, and the statisticians. The statisticians are those that do the mathematical computation to ascertain the outcome. They don't know what they are, what is the active molecule. So we go to the next. So further random control trial, we look at defined population. And so I think we've done this last week. So we've done this last week. So let's look at the advantages and disadvantages of randomized trial. Okay, we've done this last week. So let's start here. Yeah, so this is where we're starting today. So in randomized control trial is an epidemiological experiment where evaluate new preventive therapeutic. So in randomized that have maybe what we call a placebo. A placebo may be starch, normal starch or sugar or whatsoever, but that doesn't have a biological effect. So that's what you give to the control group. So you are given, let's say when you are trying paracetamol, you're trying to find out an analgesic drug, an anti-inflammatory drug. So you give to a group of people that have that inflammation. So you give them the active molecule you are working on. Then to another group of people, you can now give them starch. Okay, you can now give them starch. You know, that starch is just to measure to see whether at the end of the day there'll be a significant difference between the active molecule they took and the starch the other group of people took. Okay. So the results are assessed by comparing the outcome in the treatment group with that of the control group. So let's use an example for a hypertensive drug like amlodipine. So now in the result, you are looking at, does this drug reduce the blood pressure? In the treatment group, does it reduce the blood pressure? So assuming that you expect the drug to reduce the blood pressure by 20 milliliter mercury, then you give the drug, then you give uh, a starch to another group that is not an active molecule. So you give that starch. At the end of the day, you are going to measure the blood pressure of all the people that were in the group that you gave the active molecule. Then you measure the blood pressure of all the people that were in the group that you gave starch. At the end of the day, you can now say, okay, based on the outcome, we can conclude that this molecule, this substance we gave, actually reduces the blood pressure. So randomized control trial is considered as the gold standard among epidemiological studies in terms of validity. So when you look at epidemiology studies, randomized control trial is believed to give the best result. Okay, because you know the chances of the trial being manipulated is a little bit limited. So unlike in observational studies, randomized control trial allowed the investigator high degree of control over the study subject and conditions. Then again, randomized control trial with double blinding is of high validity. So what we mean is this. Yes, that the people that are the investigators have control, they determine what to give, but when there is double blinding, there is a high degree of validity. When the person giving the drug doesn't know what he's given, and when the people taking it don't know what they are taking, so there is less likelihood for bias. Okay, there's less likelihood for this person to come and say, oh, this is the outcome, when it was not the outcome. So the people just measure based on what they see, not based on the pre-information they have in their mind or based on what another person told them. So allocate application of randomized control trial. So where is it used? Number one, in evaluation of treatment. When there is a chemotherapy, when there's a surgical procedure, when there's any of that treatment process is used. Again, it can also be used in prevention when you're looking at prevention. So when you're looking at chemoprophylaxis, okay, chemoprophylaxis means preventing a disease or giving something to prevent a disease. That's when it's chemical based, a chemical wise, okay. So that's chemoprophylaxis. Then there is immunoprophylaxis. Immunoprophylaxis is when you use immunological substance, okay, to prevent a disease condition. Maybe you inject immunoglobulin or you inject another immunoglobulin, maybe IgG, maybe IgM or IgH, any of them, you are giving it to prevent a disease condition. So that is called immunoprophylaxis. Then there's also evaluation. You can use that randomized control trial to evaluate health program. Okay, so you are looking at a health program like you are looking at, okay, we want to check whether vaccination is right in this place, whether they are vaccinated appropriately. You can use it. You can also use it to evaluate healthcare services. You can use it to evaluate patient education program. You can use it for administrative strategies. So these are the areas that you can apply randomized control trial. So let's look at the different phases in clinical trial. There are different phases before you make a decision about a drug. you take those drugs through phase 1 trial, phase 2 trial, phase 3 trial. There's what we call phase 4 trial. There's also what we call post licensing trial. So in post licensing trial, when you have allowed the drug to be available in the market and people are taking it, everybody's buying it, you can now do a trial to check whether there are long-term or unwanted effects coming from that drug. Okay. So let's look at the different types of trials. So the first one is phase 1 studies. So what is it about phase 1 studies? So phase 1 studies is the first step in developing a drug. So when you want to develop a drug molecule, is the first step, is the first trial. Okay, where you say, okay, I think this drug is working, then you apply what we call phase 1 study clinical trial. So what happened in phase 1 clinical trial? Okay, so we say it's the first step in developing a drug. Okay, so just a minute. So we say it's the first step of developing a drug. The first step in developing a drug is to understand. The first thing is to what? How well is this drug tolerated? Okay, is it something that is toxic to people? Is it something that can kill people? So in phase 1 trial, you are looking at this drug. What is the negative effect on human being? So what you do is you look at just a small number of the volunteers. You know, in trial right now, they are doing trials for vaccines for COVID. There are people that have volunteered. Okay, there are people that say, okay, I think let me be available, test this vaccine on me. So in phase 1 trial, you just take very few people and give them that drug. Although it does not qualify to be called a clinical trial, this phase is commonly known as phase 1 trial. Take note, phase 1 trial is looking at the toxicity. How tolerable is this drug? Will the drug cause people to have increased blood pressure? Will the drug cause people to lose their sight? Will the drug kill people? Okay. The objective of every drug is to have a treatment, a good effect. But there are some drugs, okay, that causes adverse effects. Drugs are chemical. There are some chemical as little as the size of paracetamol, they can kill a human being. So that's why this trial is important. So in this phase, estimation is done to find out how large a dose can be given before a person experiences unacceptable toxicity. So we are looking at what dose. That's if you check drugs, you see this is 50 milligrams, this is 60 milligrams, this is 100 milligrams. Okay, now those are different active principle of that drug in that tablet. When you take paracetamol 500 milligrams, it doesn't mean that that whole tablet is all paracetamol and that whole tablet weighs 500 milligrams. No. When you look at paracetamol and it's 500 milligrams, it simply means that the active molecule, which is acetaminophen, can be 500 milligrams. But the total weight of that drug can be 2 grams. Why? Because sometimes the milligram of the active molecule is so small that you cannot even formulate it to give somebody to drink. So what you do is you use starch. Most times we use starch. They are called excipients. Most of the things you see, those white things may be starch. Then you now diffuse the active drug inside the starch. So when the person swallows that starch, then it disintegrates, then the active drug will now start going into the blood cells. Okay. So now in that phase 1 trial, you want to know how many milligram, how many microgram, how many SI units, okay, how many units of this molecule can be tolerated by human being. What is the highest level a person can take without causing so much damage to the body. So that is phase 1 trial. So this is called, that's we talked about, that you look at the minimum, the maximum tolerable dose. This is called what? MTD, maximal tolerable dose. So let's look at phase 2 trial. After phase 1 trial, you move to the next level. The next step is to see if that drug has biological activity or effect. Then you now check the rate of adverse reaction. Okay, so the first phase, you are looking at what is the minimal maximum tolerable dose. Then the second phase, you are looking at, okay, they say that this drug is taking care of this disease condition. You want to really find out, is it really doing what it say it is doing? Okay, and this may be why that when you look at WHO trying to argue that the COVID drug from Madagascar, they should do trial about it. You know, sometimes people that are not knowledgeable will say, hey, WHO, they hate Africa, let Africa, this is. No, there are standards in clinical evaluation. The question is that COVID molecule, has it gone through phase 1? Do we have data to support that people can take it without adverse effects? Number 2, has it gone through phase 2? Are there replicable, provable ways to show that this drug can cure people from COVID? So these are scientific things. We are scientists. You that is talking to me, a scientist, I'm a scientist. So things should be discussed in logical, rational, okay, experimental format, not just to make general conclusion and assumption. No. So in phase 2, you now take that drug. Okay, is this drug producing the effect? Is this drug really curing COVID? Then after that, you say, okay, now among the 5,000 people that were involved in this trial, how many of them are having adverse effect? You are going to evaluate that. Okay, there's a drug they call thalidomide. I remember thalidomide. That thalidomide, when it went through all the trials, it was okay. But after a long period of time, they found out that it has teratogenic effect. What is teratogenic effect? Teratogenic effect means negative effect on children that are not born. So all the people that took that thalidomide, when the children were born, they were not having hands. Their legs were cut. Do you get it? So when I remember this, I just remember it to remind you that after a drug has gone through all the trial, after a drug has been licensed for general human usage, there is also need to do a post-licensing trial to evaluate whether there are some effects that were not observed during the clinical trial of that particular drug. Okay, so phase 2, we've looked at phase 2. Then the next thing is phase 3. So in phase 3 studies, what are we doing in phase 3 studies? We say phase 1, we are looking at the maximal tolerable dose. Phase 2, we are looking at biological effectiveness or biological activity in addition with the adverse effect on the population. What percentage of the population have adverse effect? Then from there, we move to phase 3 clinical trial. Just a minute. So we move to phase 3 clinical trial. Phase 3 clinical trial is termed clinical trial in reality. That is where we do the extensive study. So we say generally designed to assess the effectiveness of new intervention and this role in clinical practice. So, okay, are we done? Okay. So in phase 3 clinical trial, just a minute, I'm changing position. In phase 3 clinical trial, so we are really evaluating okay, okay. Yeah. Okay, so in phase 3 clinical trial, we are actually evaluating what is the role of this molecule in clinical practice. Is this molecule actually doing anything in clinical practice? Is it actually making any difference? Is it something better than what we have, or is just one of those things? So that's what is done in phase 3 clinical practice. Okay, and in sometimes this phase is actually called the real clinical trial, because it's in this phase we're able to formulate policies about some important things about the trial. Okay, so after phase 3 clinical trial, and we say phase 3, we actually use randomized control trial. In phase 1 and phase 2, you may not really use randomized. Okay, you may just use a simple trial format. But when you get to phase 3, you must do a randomized control trial. Okay, because now we are going to claim that this drug is having a clinical effect, this drug is making a clinical impact. So if we are going to claim that, then we need an extensive clinical assessment to be able to come into that conclusion. So phase 3 clinical trial again is where we assess the clinical relevance of a molecule. So after phase 3, we go to phase 4, clinical studies. Okay, okay. Mm. So you go to phase 4. Okay. I'm the one that was suffer because I was stop everybody. I said I will stop everybody on the road because coffee was wrong and coffee. Yeah. So this phase is designed to assess the effectiveness of the drug molecule. Okay. So phase 4 clinical trial. So in phase 4 clinical trial, you're looking at a long-term in surveillance. So now you are saying this molecule is producing the desired effect, but you need to do what? Long-term surveillance. In some cases, it's seen as an extension of the phase 3 clinical trial. Okay, so you are looking at long-term surveillance on a control group that we are not involved. Okay. Then after that, you look at a phase 5 clinical trial. So in phase 5 clinical trial, you are looking at the efficacy of the drug, okay, which are not accidentally discovered. So this is done to ensure that the discovery is not just by accident. Okay. Design. How do you design a randomized control study? Because when you get a product out, before product is approved by the regulatory bodies, you yeah. Okay. Before a product is approved by the regulatory bodies, you need to look at all those assessments. Okay, you need to look at what is the study design. In short, even sometimes when a product has been approved and you want to convince a doctor, okay, to prescribe a product, some doctors may ask you what is the study design, because the study design will tell you the power of the trial. Okay, so if it's a design that a clinical trial where you have 200 people participating in that trial, you can't compare it with you have 200 people, 200 patients that the drug was used for. You can't compare it with a clinical trial that you have 5,000 people. Okay, that means that 5,000 you have a wider group of people that were involved. Okay, so the result, the outcome of that trial definitely will be more. It will be a stronger outcome as compared with that one which you have very few people involved in that trial. So let's just look at the study design. How do you design your randomized control trial? So the first thing is you draw up a protocol. Okay, so you draw up a protocol. This is the protocol that we are going to use for this clinical trial. There you look at your selection reference. Okay, what are you using to make selection of the treatment group and the study group? Remember we said earlier on that in your selection that the demography should be the same. Okay, if you are doing a clinical trial, the demography of the treatment group and the demography of the study group should be the same. So if you want to do a clinical trial on a new product, okay, let's say anti-diabetic product, okay, you can't say, okay, the people in my treatment group are Caucasians, then the people in my control group, they are people in my treatment group, their age is 65 and above. Then the people in my control group, in study design, you are going to look at what is your selection reference. Okay, so you are going to state that. Then after your selection reference, and then you look at the experimental population. What is the population that are going to participate in the trial? Most time it's advisable that when you're doing a trial, um, as much as possible, ensure that you have Hispanic, Caucasians, Africans. Do you get it? Ensure that at least you have so it's advisable as much as possible to ensure that your population is a good representation. Look at of what happens in global and in natural. Then you look at randomize. That's the next thing you're going to look at. Okay, who and who will be in this study group? Who and who will be in the control group? Then the next thing you're going to look at is what is your manipulation or intervention strategy? There are times the outcome may be invariance, okay, especially when it comes to adverse effect, adverse reaction. When we have variance in outcome, what is your intervention or manipulation structure? Those has to be defined, okay, before you start the clinical trial. Then the next thing you need to look at is what is your follow-up plan? Okay, so the follow-up plan is that when these patients take this drug, how do we follow up? How do we follow up to measure the blood pressure? How do we follow up to measure, let's say, the circulating hormone in the body? How do we follow up to measure? So you need to define that. Then again, you also need to define the assessment of your outcome. Okay. So after the study design, so this is just an example. Okay, this is just an example of the design, the protocol design. So we look at select suitable population. We look at sample. we make a necessary exclusions. Okay. What is necessary exclusions all about? In necessary exclusions, you can say, okay, since this drug, this is the therapeutic effect we expect from this drug. People that have this disease condition, that drug will exacerbate their disease condition. That means the drug can worsen their decision, their disease condition. These people have to be excluded. So you need to be clear about your exclusion criteria. Okay. Then the next thing we look at is who are those that are not eligible. Then who those that did not give their consent. Again, there are some people that may be eligible, but at the end of the day, they will not give their consent, or family may not give a consent. You need to put all those things in perspective in your study design. As we said, in randomized clinical trial, you look at two groups. So you look at the randomized, the intervention group, or also called the treatment group. Then you look at the control group. Okay. You have your follow-up system, you have your assessment system. Okay. Then let's look at the reference of the target population. Okay, so the regular population is the population to which the funding of the trial, um, if it's found successful, we see it as applicable. So we are talking about the population that is going to use the molecule. Okay, so it may be broad, it can be geographically limited. Okay, but with the globalization, and we are having today, most drugs are um, are not geographically limited. You see that a lot of drug cut across many countries. Okay, except when the disease condition is geographically, like malaria now. Malaria, is not in the US. Okay, Lassa fever is not common there. So a molecule that, if you have a molecule and you want to use it in the US, and your target, you have a malaria molecule, you want to use it in the US, that may not be the population. But generally, when you are doing clinical trial, we look at comprehensive population. Okay. So finally, you look at the study population. Okay. We say that the study population is derived from the reference population. So under the reference population, you now have the study population. So it's the population that actually participate in the experimental study. Okay. So we can say this is the reference population, but it's not everybody that will participate. So that number that participate is the study population. Okay. So it should have the same characteristics with the reference population. Okay. So if you have any question, any query, any comment, uh, let's look at that. So for the next 5 minutes, I'll just take your questions or your comments, and we will round up. Okay. So any question, any comment? Okay, so um, I think we don't have questions. Okay, so if there's any questions or comments, you can send to my email. If there are things we need to clarify, you can share it with my email. Okay. Goodbye from my end until we meet again. Have a great day and keep safe.